One of the fascinating ways that artificial intelligence is really shaping what is happening in modern medicine involves computational biology. And it's changing the game for what we're doing and the innovations in it, particularly surrounding some of the research and work that is going on at Carnegie Mellon University. I want to introduce you right away to Hossein Mohimani. Hossein, thank you so much for taking the time to speak to us today.
Thank you for having me.
Your work in computational biology as part of CMU's School of Computer Science is fascinating. How did you land yourself in this job? What was your path, your journey?
Yeah, so I started my PhD in 2008 with Dr. Pavel Pevsner, who is a faculty at Computer Science Department at University of California, San Diego. And that's how everything started. So at that point, Pavel is very well known for his contributions to sequencing of genome sequencing, and he just started getting interested in discovering novel antibiotics at the same time that I joined his lab. And since then, I have been working on this area, like using algorithms and machine learning to facilitate discovering of novel antibiotics. So I did my PhD there, then I was working at Illumina, which is a genome sequencing company located in San Diego for 2 years. And then after that, I felt that I really want to come back to the area that I started my PhD with, which is antibiotic discovery. And then I decided the next right move is to basically coming back to academia and getting basically faculty position. And then that's how this started.
Did you always know you wanted to work in medicine?
So yes, since I was in high school, I really was fascinated by application of algorithms and computational science in discovering novel drugs.
Now, the question that I'll yield the floor to you for a while because it definitely takes context to understand. You've talked about working to discover novel compounds so you can create new antibiotics because we are struggling to stay ahead of antibiotic resistance. Tell me about your research specifically because it is fascinating.
Yeah, so since the discovery of penicillin, a lot of very well-known drugs that we are using in our medicine have been discovered accidentally. So the story of penicillin is very fascinating. So Alexander Fleming left a petri dish with a deadly pathogen, Staphylococcus unprotected in his lab, and then he left. And then when he came back after holidays, he noticed that there are some molds in the petri dish, and the mold is killing the deadly pathogen. So he got really excited, and then he investigated what this mold is, and he was able to isolate a molecule that we call penicillin that can kill a lot of deadly pathogens. So ever since then, a lot of like best-selling drugs that we have in the market have been discovered in like really accidental ways. And what I'm passionate about is making this process really systematic and not relying on luck. And that is really crucial because right now a lot of pathogens are developing resistance against the antibiotics that we currently have available to us. And we really need discovering molecules that have novel mechanisms of action. And by doing this systematic and in high throughput, we can introduce a lot of lead molecules in the early stages of clinical setting, and that would help in basically selecting more optimal lead molecules to push in clinical pipeline, and that can really improve the efficiency of drug discovery.
I'll ask you to rewind that, and I'll set it up in the form of a question, because what I hear you saying is we can continue to rediscover penicillin accidentally over and over again, or we can very specifically, using AI and machine learning, target new compounds for the new penicillin. So how does this process work? And I know we're getting in the weeds now, which is what I want. How does this process work?
Yes, so a lot of the drugs that are currently available in the market are natural products, which means they are produced by natural organisms like microbes and plants. And natural products are really effective because they are really diverse, and they typically have chemistries that are unique to them, and you cannot find them anywhere else. And that is why they are so exciting, because new chemistry means new mechanism of action. So a lot of what has been done in the past has been like very accidental discovery of molecules that turn out to be really, really interesting. Like people would focus on like one area, one specific microbe, and just try to see if they can isolate something out of it that would be like killing, let's say, pathogens that, let's say, penicillin cannot kill. So we really want to use AI and machine learning and computational analysis to make this process like very high throughput and systematic. So we have been working with the US Department of Agriculture, and we have got 1,500 microbial strains that are very diverse from them. And then we have started collecting various type of data. This data, like we are collecting genomics data, which tells us what is the DNA sequence of this microbe and what sort of molecules this microbe is capable of producing. And then we also collect metabolomics data in cultures of these microbes, which tells us what are the chemical structures that this microbe can potentially produce. It's a fingerprint of the chemistry produced by these microbes. And then we have developed these AI algorithms that integrate genomics with metabolomics and tells us what are all the molecules produced by each of these organisms that contain novel chemical structures, that have chemical substructures that have not been seen before in any other known molecules. And then we use machine learning methods to very quickly screen these molecules to see if their mechanism of action is potentially known or not. And then for the ones that have novel mechanism of action and novel chemistries, then we can prioritize those molecules and then do further studies to see what pathogens they can inhibit.
All those processes that you just outlined up until the last sentence that you said would take decades.
Yes, that is true. With very traditional methods, it would take decades, but we have developed like these really high throughput strategies where we are able to analyze large number of microbial strains together and then process the huge amount of data that is produced in these experiments.
One of the staffers at CMU wrote a phenomenal line about the work that you do, saying if you're trying to find a needle in a haystack, you better understand what hay looks like. And what I love about that analogy and the way that you have described your work is casting a bright light into the hay that very quickly identifies where that needle is. It does not take us to the final product, but it gives us something to extract and work with. Can you take me back to the moment with your team where you said, you know what we should do? Because this had its foundation somewhere. So take me back to the beginning when you realized that AI and ML could help you accomplish this and solve for this problem.
When you look at the publications in this area, you would see that each paper is reporting, let's say, one novel molecule, which is exciting, and what are the bioactivities of these molecules. And I remember a meeting that we had in our lab where we were saying there are a lot of other fields that are becoming really high throughput, and we need to get away from this process of like spending years on a single molecule and make it really high throughput, where in the future when people work on this, they would work on experiments out of which they will discover thousands or tens of thousands of molecules. And we really wanted to make this possibility, and we wanted to show some proof of concepts that this is possible. And we are still, we have already shown some proof of concept with the initial data that we have, but we really want to move like far beyond this and make it completely systematic and high throughput process from the beginning, when we start from, let's say, the organisms, the natural organisms, all the way to where we find the bioactivity of the molecules.
Let's talk about some of that proof of concept work. I have seen at least two specific instances where you've used algorithms successfully. Can you walk me through those? One with I believe it's PNPs, and the other specifically with the Candida fungus. Let's start at dealer's discretion, whatever you would like to discuss first.
We looked at 1,500 microbial strains that we received from US Department of Agriculture. We collected data on them. We analyzed them using the algorithms and machine learning methods that we have developed in our lab. And we were able to find hundreds of novel peptide natural products, PNPs, in these samples. And then we were looking into them to see which ones are likely to have novel chemistries and novel mechanism of actions. And we do that by using machine learning methods that look at the substructure of these molecules and figure out whether they correspond to known mechanism of actions that are available in other molecules or if they are novel. And then we prioritized a few of those molecules, and then we isolate one of them and did in vitro testing against deadly pathogens. And we realized that this molecule is capable of killing Candida auris. So Candida auris is a deadly and quickly spreading pathogen that has been classified as urgent treat by Center for Disease Control. Right now, there are variants of Candida auris that cannot be killed by any of the antifungals that are currently available in the market. So this is really scary. And then we collaborated with National Institute for Allergy and Infectious Disease, who they got really interested in this molecule because it could kill pathogens that they knew none of the other existing molecules can kill. And then they decided that they want to collaborate with us and test this molecule in vivo in mice. So the tests show that the molecule is completely safe in mice, and at the same time, it is very effective. It would significantly reduce the burden of fungal pathogen in mice kidney. So that was really exciting, and that enabled like next steps, which are pharmacokinetics, and after that, IND enabling studies. So we are going on, we are going down through our lists and isolating. We have so far isolated 4 more molecules, and we are going down the list to isolate these interesting molecules and figure out what are their bioactivities.
Are the other 4 molecules also related to antibiotic resistance or possibly cancer and other mechanisms where discovery like this may be applicable?
What is interesting about natural products is that they have very diverse substructures, very diverse chemistries. And this means that they can have very diverse mechanism of actions. Some of them are anti-cancer, some of them are antifungal, antibacterial. So we have some predictions about potential activity of these molecules that these molecules could have, but we have not tested them yet. So we are interested in all of those areas.
What was a substantial challenge that you had to overcome in this work?
Yeah, so one of the biggest challenges that I wouldn't claim that we have overcome it, I think it's still challenging, but we have maybe taken one baby step toward it, is making the data available. So right now, like I think we conducted one of the, when we looked at 1,500 strains of diverse microbes, we conducted one of the largest data collection studies in this field. And that data was the crucial factor that enabled the discoveries. And also it sort of enabled improvement of the methods, because what is we not only we need like really sophisticated AI and machine learning models, but on top of that, we also need really high quality data, training data, to feed these models and enable new discoveries. And I think this is like a very important point. Nowadays, machine learning and AI is getting so advanced that people might get confused that we don't need data anymore and we can just start running these models and make discoveries. That is not true. And these models are going to be helpful only if they are fed with really high quality training data. So I think we have taken the first baby step toward that, but we probably need to collect a lot more data to really realize the advantages that these fancy and sophisticated machine learning models can give us. So that, I think, was one crucial step beside like the development of algorithms and machine learning models that enable this study. Other approaches to this problem that are really exciting, like for example, discovering what is the target of natural products or drugs in general, because that can give us a lot of information about what is the mechanism of action of the molecule, and that can enable discovering molecules that have a specific protein that can inhibit a specific protein target. But that is also a very challenging task, especially because, again, there are not a lot of training data available that can feed the machine learning models. So in that area, what I'm really interested in is some of the works that has been done on high throughput methods. And I'm talking about wet lab methods that can generate data on interaction of small molecules with protein.
Congratulations on your success there. That must have been a wonderful moment.
Yes, and this was thanks to amazing PhD students that I have been working with at Carnegie Mellon.
When you look at these students you're working with at CMU and you're thinking about where you want your own research to be 5 years from now, what direction are you hoping to go? I realize you're still engaged, but what's next?
Yes, so again, if we really want to pick the fruits of all the sophisticated artificial intelligence and machine learning methods that have been discovered in the past, I think there are two challenges. One challenge is data that I already talked about. And the other challenge is what is called like being able to generalize to out of distribution data. So that what that means is, let's say, for example, in image recognition, people have data on dogs and cats, and then they want to classify if a new image is dog or cat. And they do that with really high accuracy, and that is exciting. And that is basically what people want. But when we come to drug discovery, small molecule discovery, what people really care about is discovering molecules with novel mechanism of action and novel chemistries. So if, let's say, we have data on penicillin, and then we develop a method that identify penicillin or identify a completely novel molecule, but with a mechanism of action like penicillin, then no one is gonna get excited, because that's what we really cared about was molecules that have really novel mechanism of action. So right now we are using machine learning mostly as a way to identify what has known mechanism of action, and then paving the path for and then focusing on the rest, which don't have any known mechanism of action, which means potentially they have novel mechanism of action. But what would be really exciting here is coming up with methods that can truly go beyond this and enable finding novel mechanism of actions. And that is something that has not happened so far, but it would be really, really exciting. And that is something that my lab will be working on.
What support is necessary for you to accomplish that goal? I imagine it's maybe not one of compute power, But perhaps it is. Is it compute power? Is it funding? What is it that has to happen in this pipeline for you to fully embrace and lean into that?
So, in a lot of fields, machine learning started becoming efficient, becoming accurate when the size of high quality data uh, grew more than some threshold. So in this field, at least so far, and I also believe in the future, I don't think computational power is going to become a bottleneck. And I think the bottleneck again is the sort of data that we need. And in this case, it could be like a lot of molecules and their known mechanism of action. And probably, nobody knows, but probably when the size of this data uh, grow and get larger than some threshold, then maybe many of the existing machine learning models start becoming accurate.
Hossein Mohimani, thank you so much for your time and perspective on this conversation about what AI and machine learning can do in modern medicine.
Thanks a lot. Thank you for your time, and thank you for having me.
You've been listening to Does Compute, the podcast that examines how computer science is building useful stuff that works. Thanks so much for joining us. I'm Steph Strickland.